Hello, hello. I think we've got it. Let's just double check. Ari, how you doing?
Hello, hi, there's two of us. It's so fun.
Woo!
Welcome, everyone.
I'm super excited. I know who I am, so I'll introduce myself, and then I'll let you introduce yourself, Ari. But my name's Eli Schwartz. I've been teaching the MCAT since 2005, specifically here at Kaplan. And just super excited to go through a lot of the content in the background. Most of what I do right now is I do a lot of these sort of sessions. So these kind of pain point, really focused sessions about very high yield topics like metabolism and private tutoring. So I do a lot of the tutoring packages that people get through Kaplan, in addition to their class. That's Ari, do you want to give yourself a quick intro for your background as well?
Sure. So my name is Ari Kim. I am also a Kaplan instructor, mainly dealing with MCAT. I've been teaching online for a good number of years now. I teach in classrooms. I also tutor. I am also on the channel. And I also do content development work in the background. So this is my chance to share the joy of a topic that I think a lot of students struggle with, which is the topic of the day. It's going to be metabolism here.
Yeah, Ari is amazing. We're super excited to teach this session together, as not the least of which because I know that I get to watch Ari teach for the first half or so of our session. I can chime in and ask questions. I kind of pose as a student for a little bit and get behind the scenes on what she's thinking, but also just because it's so much fun to hang out and teach these topics together. And metabolism is absolutely a super high yield topic. Something that if you can get a lot of the fundamental building blocks of, in this session in particular, as we go through glycolysis and citric acid cycle, it will absolutely feed forward into some of the other major systems and processes, especially tracking energy movement and tracking that cellular metabolism comes up super frequently, even on sort of fundamental foundation questions above and beyond the larger scale experimental stuff. I've been talking for a little bit. Ari, do we want to jump right in and start tackling it?
Here we go.
Let's
So
do it.
you should be able to see my picture in the corner and the big blue background that says metabolism, glycolysis, and citric acid cycle. And that's going to be the focus of our session today. Now, I'd love to hear from you, Eli. What kind of feedback do you get from students when you tell them today we're going to sit down and do some metabolism? What kind of like usual responses do you get?
Well, never any eye rolls. Everyone's always excited to memorize more citric acid cycle. Uh, but I think the big questions that I always get are how much do I actually have to memorize? And and I guess maybe a secret question that's in that is not just like how much do I have to memorize, but do I have to memorize? Like, is there some way I could think about this a little bit differently than I did in my 101 undergrad class or my bio class in high school maybe that went through this briefly? How can I'll use air quotes, understand it a little bit in a more rich fashion outside of just memorization. So I think that's the big thing I'm looking forward for at least glycolysis.
Right. And I think one of the things that I remember struggling with when I was in school as well is, okay, I got to sit down, I got to remember the steps, I got to remember the enzymes, I got to remember the intermediates, and it becomes really a lot. And one thing that both Eli and I have in previous kind of conversations have discussed is understanding how to learn in layers. I love the idea of learning in layers. And this does require a sense of repetition. Like you're going to head back to this. So I don't want you to feel like after the hour and a half with us today, you're going to be like a total master of metabolism with glycolysis and citric acid cycle. But I want you to understand that there is a sense of foundation, that you have a sense of organization of how this all fits together. Connecting the dots is the biggest thing in both metabolism as well as just learning the science on the MCAT as well. So that's what we're going to do. We're going to try to help you understand how to learn these aspects of metabolism and layers. And really, like, what the amazing takeaway about that is if you understand the sense of how we would approach it here, you can go forward. You could talk about the ETC. You could go and do glycogenolysis. All of the other kind of related aspects of metabolism you can approach in the same sort of way. That's really my wish for you all here, is to show you, and I'm excited to share this with you because I struggled with this so much when I was in school as well. So let's get started. We're going to kind of break this into two. We're going to do glycolysis, citric acid cycle. So obviously we're going to start with our learning objectives here. We're going to talk about the function of very key enzymes in glycolysis. And in the second half, we're going to transition on over and describe some of the components of the citric acid cycle. So here it is, boom, nutshell. This is glycolysis, all 10 steps of glycolysis. Now, Eli, if you were somebody who is going to chunk this down, what would be some very convenient ways of chunking down glycolysis?
I think, and this is true for all of the metabolism processes area, I think what I would do first is make sure I know where energy comes in and where energy goes back out. Energy in, energy out are generally the first quick big picture things I look for a systemic level, for like a homeostasis level. If I'm lacking a whole lot of energy, where am I going to get it back out of this process? What are the sort of feed forward reactions? And if I have plenty of energy, where can I spend it? Maybe in storage or in an initial part of a process, initial part of a loop. I think that's first big picture. I always look for that. The second thing is, and it sounds a little bit silly, but remembering the name of what goes in and out sometimes gives you the enzymes for the thing. So you don't have to necessarily memorize the enzymes as a whole separate step. If you just know the order of the reagents and you know the order of the products, oftentimes you get the enzymes for free built in. So those are the two big things I always look for. How about you?
I'm gonna copy you. I think, kind of dividing it out into the energy and energy out. In fact, very conveniently, here is the energy in steps, parts of glycolysis. And then here is the energy out parts, our benefit of glycolysis. It very conveniently divides this into half. And that's actually how we're going to approach this as well, is let's talk about the input of energy into glycolysis and then also kind of see the output of it. And it's not going to be a net of zero. If you notice that there's like two things like this, it's going to actually benefit you. Glycolysis does give you some energy. So one thing here is if you have questions or if you want to talk about specific things that we discuss, I really want you to head over to that poll tab in there and go ahead and talk, because it's not just Eli and I kind of mistakenly kind of forgot to mention this, but it's not just us in the room. We are actually joined by so many other teachers in here. So go ahead and feel free to chat and have a conversation. It's one of the best benefits we have about having classrooms like this. Come talk to Owen and AJ and Carrie and Nick. They'll be more than happy to have conversations with you specifically about any questions you have during the session, as well as just overall if you have questions about your prep or anything like that. So yeah, going on back over to this, it does divide it very nicely. And then, Eli also talked about the freebies. And I think I love that aspect about biochemistry in general, is that the enzymes are named very, very logically. So there's two parts to name, usually two parts to that name. The first thing is usually going to be what goes into it. Right? And then the second part of that name is usually the function, like what kind of reaction, what kind of enzyme, or what kind of reaction this is describing. So if you ever see the word kinase, for instance, what would you know about that, Eli? If I told you something was a kinase, what would you then say?
I love these sessions because usually, so Aries, of course, posing this to me because as she mentioned just a second ago, there's a lot of opportunity for you to reach out to other instructors, but not directly interacting and asking questions to us like we normally would have, just because of the way it's streaming today. But a kinase is usually something that catalyzes a transfer of a phosphate group. So from high energy phosphate donating things to specific substrates. And usually we would talk about a kinase in the sense of a phosphorylation reaction. So that's, it's just a nice question for us to pose out to students. So if you heard that question just now and went, oh, I think I know the answer to that as well, say it out loud to yourself, throw it down into the poll chat. That goes to some of our off-camera MCAT experts. As already mentioned, this is a chance for you to interact as well, even though we're not going to see your immediate question. 4 or 5 other MCAT experts with probably just as much experience are going to grab those and answer them as well. So long answer to it, Harry, but that's my answer for kinases.
Right. And I think this is our way to have the interaction. We're going to essentially kind of go back and forth and have conversations about this, posing questions that we would normally kind of adjust to you. But I want you to kind of think that we're asking you guys as well. And if you want to answer them, please do feel free to do so. Okay. Okay. So, so the other kind of like very common enzyme I see is dehydrogenase. So if you ever think about dehydrogenases are types of it's part of the enzyme in the oxidoreductase class. And oxidoreductase, oxidation reduction, talks about the movement of electrons. So one of the key things that we want to look out for in metabolism is the movement of those electrons, because ultimately that's what we do. When we take, carbohydrates and we reduce them all the way down to energy, what we're doing is stealing the electrons from that, and then we're going to funnel that into creating ATP out way at the end. So so those are just some like handy tips, because what I understand and I see comments kind of coming in is, do I have to sit here and memorize this whole thing? You could. It would be testable, right? But is that the best usage of your time right now, especially if you're relatively new to metabolism? Probably not. One of the things I like to share with my students is the proportion of how things are tested. And if you have done any questions on metabolism, a lot of them don't tend to ask about very specific steps or enzymes, intermediate structures. They ask about, how does this all fit together? That's going to give you, like, I don't know, I want to ballpark a number. Like 3/4 of the questions will be big picture. How does glycolysis funnel into TCA? How does glycogenolysis, help me when I am, when I'm hungry? Like things like that. Big picture concepts always should be learned first. So what I would love to know from you all, and I want you to think about this right now, is instead of like looking at this very kind of intimidating diagram, I want you to think net. What goes into glycolysis? What is the thing that funnels in? What is the very first compound in glycolysis? If you're thinking glucose, great, that's right. Glucose is the thing that goes in. What do we gain from glycolysis? Okay, I chop it in two. What do we call that? We call that pyruvate. I get a little bit of energy from that. Perfect. I get a little bit of an electron carrier. Great. That is the net reaction for glycolysis. And really, that's like 3/4 of the questions. This is what you want to understand is, what is the net reaction? And then where does it occur? So glycolysis, being a part of anaerobic respiration, is actually outside of the mitochondria. This occurs in the cytosol of your cell. And that is another key aspect that I would want you to do, okay? So what we're going to do here is focus on the chunks. Focus on the chunks. So Eli a little bit earlier talked about energy in versus energy out. We're going to talk about the energy in aspect of glycolysis. So the first step is going to be the very first enzyme or the very first reaction in glycolysis, this one right here. So this is part of what we call the preparatory phase. We're going to prep our glucose in order to get some stuff out of it afterwards. So to Eli's earlier point of how enzymes are named very logically, the very first enzyme in glycolysis is called hexokinase. Now you're like, okay, fine, another thing I got to memorize. But if you wanted to maybe logic your way through this, we could break down the word hexokinase.
I love logicking, Ari. I'm so excited.
Okay, so think about this. The prefix hex. What does the hex mean? Well, hex means the number 6. What do you think, Eli, if I was to pose you as student Eli here, what do you think this hex is referring to?
I think it's a 6th something. It's either a 6-member sugar or a 6th position.
It's both. Ta-da, it's both. So, and I know that, I'm not asking everyone to memorize the structures of all these, but personally, when I was learning how to do this, and not just like, I got to cram all this stuff in my head, but when I was really getting into the weeds of really understanding what's going on in glycolysis, the thing that helped me the most was actually the structure of those intermediates. Now, I'm not saying you need to go memorize this. I'm just saying this was the logical way for me to understand what's going on. So to help you understand what I mean by this, I'm going to quickly just sketch out what this looks like. So CH2OH, here is glucose. And yes, you do want to know the structure of glucose.
That's a good point. I was going to ask you that in the last slide, Ari, but glucose is so commonly tested. The ring form in particular is always nice to see for things like glycogen and storage, but also if we break it apart and we take a Fisher projection of it, always nice to have that as well, so we can instantly recognize glucose for some of the other isomers.
Correct. Yeah. So this would be one of those carbohydrates that you do want to just have memorized. So this is my task for you. This is one of those ones you want to get known. Okay. So let's talk about this. This is glucose. Glucose comes into the cell through, GLUT4 or GLUT2 or glucose transport of some sort. And then, what's interesting about glucose is there's no problem with the cell just picking it back up and shoving it back out, because there's nothing to drive it in and out other than just the presence of it. So the cell does something really, really important in the very first step to trap it here. And that's really what's going on, is if I understand that this is changing it into glucose 6-phosphate, the hex is referring to both the fact that this is a 6-carbon sugar, but also that I'm adding it to a very certain position, which is essentially the 6th carbon. So again, if I was to redraw this guy, I'm going to skip the OHs because they're mostly unimportant for this conversation, right? I'm going to add in a big old phosphate, and check it out. Phosphates have negative charge, and anything that has a charge is going to be much harder to move through across any cellular membrane, right? Even though it's in this transporter, now I've locked it inside the cell. So it can't pick it back up, it can't lose it. We've done all this work to put it in, and now it's trapped right here. So we've added this negative charge, and this is our hexokinase that's adding the phosphate onto our glucose, trapping inside the cell. So that's the first step. And we know that this is going to happen. If you knew the enzyme, you're like, I'm not 100% sure what's going on. Hexokinase is a great reminder. A kinase is, like Eli mentioned a bit earlier, grabbing a phosphate from an ATP molecule and adding it onto the substrate here. Okay.
Now I'm concerned about something, Ari. It says that ATP had to be used, and that makes good sense to me. ATP to ADP, triphosphate to diphosphate. That's how we got a phosphate group on here. Is that something that can drop back off? Can glucose drop it back onto ATP if it wanted to?
Ooh, let's think about that. That's a great question. If I think about the way that ATP is structured, ATP, adenosine triphosphate, everyone knows it's like the energy currency of the cell, but it's also in the name itself, tells you a little bit about the way it's constructed. ATP, adenosine triphosphate, means I have 3 of these phosphate groups in series to each other. That's a lot of negative charges that I've now put in a very close proximity. This isn't really a reversible reaction. Once I grab that on and I put it on something else, that's it. So that's one of the key characteristics actually of this step, is that it is irreversible. I cannot go backwards. And that makes this step one of those really important enzyme steps. So a little bit earlier I mentioned, I would want you to know the net reaction. If you were going to go one step, one layer deeper, it would be to know these important enzyme steps. So step 1 is one of those irreversible steps, and that is one of those important steps that you do want to keep in mind. Now, Eli, why do we care about irreversible steps? So what? It doesn't go backwards. Who cares?
Well, the thing for me for irreversible steps is usually there's something pretty clear, like you very clearly mentioned, where I have 2 extra negative charges over here and only 1 here. It's never going to drop spontaneously back off and go back onto ATP. So I really want to know what that forward reaction is, because if I make too much of that product, I make that product too fast maybe, or I make that product in an energy situation that I needed something back, I now have the glucose trapped. So I want to know what the irreversible processes are that I can't go backwards if suddenly I realize, oops, I needed that glucose somewhere else.
Right. Yeah, so absolutely. It gives you a lot of indicators of modes of essentially regulation. A lot of the steps can actually go back to the specific irreversible steps like here and tightly regulate this enzyme in ways so that it can make it stop, right? Because I've got too much, or speed up because I need more of this forward process. Okay. Yeah, so this traps that glucose inside the cell. Excellent. So now I understand what hexokinase is doing. I understand, this important irreversible step. I understand the involvement, right? I need to have a phosphate somewhere. So let's take a look. Oh yeah, and then this also makes sense too, right? We know that there must be some sort of regulation in those important steps. The immediate product, glucose 6-phosphate, will actually go back and inhibit. And this is a form of feedback inhibition, right? Anything of the downstream process going back up and then inhibiting an enzyme from an earlier step is called feedback inhibition. Okay, so let's talk about the next kind of sets of processes. Again, focusing a little bit more on the big important steps in here instead of really kind of regimenting each individual step. The next important step we're going to talk about is the involvement of fructose. Now, you're like, we started with glucose. What happened? Where did we get fructose from? And again, I would encourage you to kind of take a little bit of a closer look at the structure, because glucose and fructose are very, very, very similar. So what's happening is in this next step, in the step 2 to 3, let me go back here actually. When we go from step 2 to 3, we're just reshifting things. And this is another class of enzymes that I would encourage you to all know. Anything that is involving the rearrangement of molecules, I'm not adding anything, I'm just shifting things around, is called an isomerase. So glucose 6-phosphate going to fructose 6-phosphate is one of those not super important steps. I don't have any energy in or out or electron carriers, but it is still really important because I'm going to rearrange my molecule for a preparation for the next step. And so in a little bit, I'll show you exactly why it's important to turn this into fructose. But first, I got to talk about this step. So we got fructose 6-phosphate, a similar 6-carbon sugar with a 1-phosphate group. And then I'm going to add another one, right? So I've got fructose 1,6-bisphosphate. Bis is just meaning 2. So I've got 2 phosphates in 2 different positions on this fructose molecule. So again, why? Why do we bother? What's the point of adding more phosphates here? I'm wasting valuable energy. Eli, tell me, help me. Why do I want to bother doing this?
Well, before I answer that, because I've got an answer for you. Before I answer it, I'm concerned about the 1,6. How come it's got to be 1,6? Why couldn't it be 1,4, 1,5, or something else? What's the 1,6 all about?
Well, let me show you. So again, I'm going to draw this out. I've got a phosphate here, and I've got another one. Whoops, I got a CH2 phosphate here. Both these guys have negative charges, and then I've got some OHs here and here, okay? Mm-hmm. Now notice that this is a pretty, I would say, symmetrical molecule here. But also, a little bit earlier, I talked about how phosphates are involved in ATP. Now, like, why does ATP even have that much energy associated with it? Well, I've got a bunch of negative charges in very close proximity to each other. When I have phosphates on either side of the oxygen kind of symmetrically dividing it, I've now amped up its energy. It's really, really much easier to cleave a normally very strong bond. Carbon-carbon bonds are usually quite strong. So this here is a way to prepare this molecule to cleave it a lot easier downstream, which is essentially the next step after that. So that would be my answer to you. I'm curious, is that what you were thinking?
Yeah, absolutely. That's exactly it. The further away, the better. And that's why we had to use isomerase to get fructose in the first place. You know, once we have isomerase, then we can have a single carbon off at the end that has an alcohol group that then I could put the phosphate onto and rejoin that ring around that middle oxygen. Otherwise, I couldn't have done it with glucose. I have to use isomerase to make fructose in the first place.
Great.
Yep.
Yeah, absolutely. So that's right. Yep.
And I secretly avoided answering your real question, Ari, which is, why would we want to do this? Well, I want to do this because I want to make a really high energy group. I want to have something that has so much energy that I could then break it down into smaller pieces and get the energy back off of it.
Absolutely. So this is a very, very important step. In fact, if you were to memorize one step that was like the most important step, it's actually going to be this one right here. So this is mediated by an important enzyme, phosphofructase kinase 1. Again, the name spells it all out, phosphose, fructose, and then it's a kinase. And this is what's going to add on that second high energy phosphate molecule on there, right? We obviously are going to hydrolyze ATP because this is a kinase. This is also an irreversible step, also telling you that this is an important step that can be regulated. But even more than that, the reason why this is the #1 enzyme you want to know for glycolysis is that this is a rate-limiting enzyme. Rate-limiting enzyme is another kind of way of understanding that a series of steps is always going to be the fastest from the slowest step. So I guess in better words, it means the fastest this enzyme, this entire process of glycolysis is going to occur is regulated by this one step. If this is the slowest step, this would be the one that's going to dictate the overall rate of the entire reaction. So this is an extremely important step. It is also very, very highly regulated. I do see a question coming in. I thought I'd address this as well. Is there a list of common structures, or organic chemistry structures that we have to memorize? Now, I will say I'm not a huge fan of memorization of aspects just because it tends to be very fleeting. But there are some here, and I mentioned one. Glucose is one that you do have to have memorized. If I was to think about carbohydrates, I would recommend you also know the structure of fructose and galactose. And then you want to know how they combine into disaccharides like sucrose and lactose would be another really good one. And then if I think a little bit bigger, you want to think about like how glycogen is put together, so like polysaccharides. So that's what I would recommend you do memorize. But in general, OChem structures aren't really something you want to just memorize. You want to know like functional groups and how the IUPAC nomenclature kind of dictates the way that they're all put together. So that's what I would say is know the functional groups, know how IUPAC rules work, but I wouldn't recommend just wholesale memorizing things unless we're talking specifically about carbohydrates.
And I think that's a great follow-up point, Ari, that a lot of people ask me, should I memorize D-glucose and L-glucose on separate flashcards or something like that? And, I always come back with the classic answer, which, D-glucose is the one that we really care about the most. But then if you know D-glucose, you instantly get L-glucose, right? You just flip those chirals. So it's not that you have to have everything memorized. I completely echo your point. We want to have a little bit of logic to it. The only one I would throw in there that I sometimes see come up is mannose, usually as an isomer class, but galactose, fructose, glucose, sucrose, and lactose, probably the most common four I would recommend. I'll just throw mannose on there as a fifth. And to your point about not memorizing, the rate limiting point here, the rate limiting step, it makes a lot of good sense. And I think that's a big part of getting these bigger picture cycles is trying to ask yourself, why does it make sense for this to be rate limiting? And my short answer is we just put 2 phosphates on this tiny little sugar. That makes a lot of good sense that this is going to be the hardest thing to create. Everything after this had better be easier to create. So this should be the rate limiting step.
Great point. Absolutely. I see another question. I love this one question. I think it ties so nicely to our discussion, Eli. I've got a bunch of questions asking, I was ignoring this on purpose, wondering if anyone else would notice. Why do we have magnesium? Why do we point out the fact that there's magnesiums? It's a cofactor for PFK1 and a lot of the other ones. And my big hint to you is I want you to think, they're very commonly associated with kinases. What is the role of divalent cations like magnesium when you are working with kinases? Well, if you think about the fact that we are now housing a bunch of very charged particles in a short kind of like span, it's to help stabilize the presence of those negative charges. So if you think you're working with a ATP or really anything that's got those kinds of charges, it would make logical sense to also have cations around to help stabilize, to be able to kind of fit them in very specific geometries in order to hydrolyze those things off. So very, very common to see in a lot of these enzymes, especially in metabolism that talks a lot about ATP.
I got a follow-up for that, Ari, and that's how come we couldn't just use sodium? How come we just couldn't use potassium? Why you got to use magnesium or manganese or calcium? Why you got to use the divalent ones?
Great point. I mean, so the monovalent, so like sodium and potassium things, they have other roles outside of this. They don't tend to be involved in processes like stabilization because it's just the one charge. When you got 2 charges, though, you have twice the power of stabilizing really complex compounds like ATP as well. So I think at a nutshell, that's how I would kind of view it. I need to have more charges around in order to stabilize more charges coming from an ATP. But I'm actually curious to see, is there something I wasn't thinking of?
No, that's exactly it. That's exactly it. And the big thing that I always put out there is the divalent cation that you're using for stabilizing, because again, you're stabilizing phosphates. You need to have that high positive charge. The divalent cation that you get to use kind of depends on the environment. So the vast majority of them are going to be magnesium like we've got here, but you can see manganese in the process that I'm going to do after this. And you can also see calcium being used, especially as a feed forward bonus. But again, divalence, you really need that extra push.
Great point. Okay, so we prepped you guys up. This is the rate limiting step, the important enzyme. It also has, and I'm not going to go into a ton of detail because this is it's it's a lot of detail. But the other kind of thing you want to think about here is also the involvement of another regulatory molecule called fructose 2,6-bisphosphate. And this is mediated by an enzyme called PFK2. And this is an enzyme specific to the liver. Now, in a nutshell, I don't want to get too into the weeds here. Liver is the place where you want to store a lot of molecules, right? It's involved in glycogenesis, right? So the storage of glucose is not for the purpose of breaking down, but because they want to kind of, put them all into big polysaccharides for storage because I got enough glucose around. So this guy is a mediatory molecule that essentially activates PFK1. And this is to essentially prevent the feedback inhibition that would normally occur. If I've got a lot of fructose 1,6-bisphosphate, normally that guy would go back and say, okay, PFK1, enough. I've got enough here. But instead, if I have this workaround specifically in the liver being the place to store glucose as glycogen, I have a way to kind of override that feedback inhibition. That's really the big role for PFK2 in the liver itself. So yeah, and then, to our points a little bit earlier, it would totally make sense that if I have lots of energy, lots of ATP, it would inhibit PFK1, whereas AMP, adenosine monophosphate, which is the energy depleted version of ATP, if I've got a lot of that around, it would activate PFK1. Okay, I'm gonna go ahead and erase some of this stuff so you guys can all see what's going on. But we've prepped up this molecule, we've made it symmetrical, we've got a phosphate on both sides, and the next step here is the cleavage part. I'm just going to chop it right in half. Now, it's not perfectly symmetrical, which is why you have two different products. You have glyceraldehyde 3-phosphate, and then you also have dihydroxyacetone. But again, if you looked at the structure of these two, they're very, very similar. And what one path can be is that we have an extra step where we can convert this dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, or it could go do other things. For example, a common alternate pathway here could be to make some glycerol. Glycerol is an important component for the backbone to make triglycerides. So it's essentially a way to kind of, I don't need enough energy, I got enough of it, so I'm going to go make some fat instead. And so this would be one of the alternate pathways, especially when you are energy full. Okay, so, right, we talked about that. And this is, again, another kind of important point to note here. I want you from this point onwards, assume we've got twice the number of glycerol 3-phosphate. Counting the carbons is actually a really good habit also in metabolism, in carbohydrate metabolism, because that will also help kind of keep track of what's going on. So I've taken 6 carbons, I've chopped it into 2, and now I've got 2 3-carbon compounds in the form of glyceraldehyde 3-phosphate. So this is where we're going to get our money. This is where we're going to go head on over to the payoff phase, because at this point, all we've done is input in energy. We do need to gain some energy now. Okay. So next step, let's take one, right? Remember, there's 2 of these guys, right? We're going to essentially do this rest of this path twice, glycerol 3-phosphate, and then we're going to involve this important molecule, NADH. And so a little bit earlier, remember, I wanted you to think, what do I know about the enzyme names? If you see the involvement of NADH, you want to think, okay, this must be a dehydrogenase. And if you're thinking it's a dehydrogenase, perfect. That's exactly what you want to do. So our next step here is a dehydrogenase. We're going to essentially couple, and this is a kind of an interesting thing. We're going to couple the movement of a phosphate onto glyceraldehyde to create 1,6-bisphosphoglycerate. And this is coupled to the generation of NADH. Now, one thing that I think I've learned a lot, especially when I consider the chemistry components of metabolism, is that things just don't come out of nowhere, right? Like this is a generation of an energy carrier. This is the reduced form of NAD. So NADH, that means it had to have taken an electron from somewhere. Where did it come from? Well, probably the process of where we added this phosphate on. So that's it. We're essentially now harvesting our electrons to be able to then make our electron carriers. This is then going to go downstream. And I see a question asking about the role of electron transport carriers have in metabolism. They're the reason why we do this. These guys are electron carriers. We harvest these electrons, and then we're not going to get a chance to talk about it today. But downstream, when you get to the ETC after the citric acid cycle, the ETC is going to grab all of those electrons, and then it's going to kind of like, essentially the way I always kind of visualize this is like a dam. It's a way of harvesting the energy, the potential from the movement of these electrons spontaneously moving down. It's going to push a bunch of protons into a specific state. That is essentially going to create a gradient that's going to be used to harvest ATP into energy as a form of ATP. So that's the big role. Electron clearers are the reason why we do this. We are essentially oxidizing carbons, generating a lot of electrons, and then making energy out of them.
That's exactly what I was going to add, Ari, to what you were already saying is, anytime you're moving protons around, think in the opposite direction you're moving electrons around. So if I've got a proton leaving something, I've got a bond to oxygen or an oxidation gained in that process, and vice versa. So yeah, the mitochondrial matrix has a lot of electrons and the intermembrane space has a lot of protons. And at the end, we're going to combine those back together. Moving protons into the intermembrane space in the electron transport chain allows us to have electrons on the other side. And that's what's giving us that high energy to allow the ATP to actually be formed. Love it.
Great. Okay. And to be clear, this isn't something that's only specific to the storage of these molecules. I did mention liver earlier and how liver was a little bit special and involved the storage of glucose in the form of glycogen. But glycolysis is something that happens really pretty much every single cell out there uses glycolysis. So this is the cytosol of those cells. So if it's got a cytoplasm, which I think really every cell has, it can go through glycolysis. This is a process that just happens all the time. So great, yeah. So now we've got our very first generation of an electron carrier. We've added a phosphate. And you might be wondering, well, how come this isn't a kinase? Well, it's not a kinase. I also had that question. Why is this not a kinase? Because it's not actually taking it from ATP. There's like an inorganic phosphate floating around here, and it's adding it to there. So it's actually kind of a part of the entire process, coupled to the generation of that NADH. So this is NADH generated. It produces this really high energy intermediate. And team, why is this high energy? Think about the chemistry. This is a high energy molecule because it's got 2 negatively charged phosphates on a 3-carbon sugar. This is actually even more high energy than what we saw a little bit earlier. So very high energy intermediate here. And interestingly, this one though is irreversible. This one is reversible. So this is one of those steps that you want to kind of think about with regulation wise. It's still a really important step though here. Okay, next step. We're going to get some payoff. We're going to get some currency here. So we're going to take our high energy intermediate, and then we're going to couple the movement of one of those phosphates off, and then we're going to make 3-phosphoglycerate. And this is it. So we've got some ATP out of this. And then also kind of thinking ahead, remember, we're going to do every one of these steps after the cleavage part twice. So it's not just one ATP we're making. From every glucose, we're actually making 2 ATPs in this one step. Okay, this is generated by the enzyme phosphoglycerate kinase. Again, you can see that little magnesium involvement here. We get some energy. This is also called substrate level phosphorylation. So I bet you've gotten this question a lot, Eli. What do you think? What is the difference between substrate level phosphorylation and the other very common term, oxidative phosphorylation?
I do get this all the time. The shortest answer I always give, and I'd love to hear if you have a longer one, is substrate phosphorylation just means it doesn't require the electron transport chain. That's the big difference here when we talk about substrate level.
I agree. So anything that is the generation of ATP that isn't ATP synthase, it's just this thing that is essentially coupled to the electron transport carrier, that is substrate level phosphorylation. So here is a good example of substrate level phosphorylation. This is another irreversible step. So it's not one of the ones that you want to think about in terms of regulation.
I have a follow-up question to that before we go on. I sometimes get that question. So if it is reversible, is it likely to reverse? Does reversible mean it's likely to go backwards, or just that it's possible?
Oh, such a good question. If you think about the process here, generating the next step onwards, this isn't just like a step that happens and it just kind of sits around and waits. Usually the requirement of the energy will essentially fuel the forward reaction that happens. So while this can go in reverse, it generally doesn't. But, and we can really get into the weeds here. Eli, what is the opposite of glycolysis?
Gosh, there's so many good answers to the opposite of glycolysis, but I would say a storage of that sugar of some kind, perhaps, Ari?
Yeah, that is one form of the opposite, right? Instead of breaking it down, I'm going to store it. But if I was to consider, well, we start with glucose in glycolysis, is there a process that is essentially going to make glucose instead? I was thinking kind of like, I don't know, gluconeogenesis,
Like a
right?
new glucose, maybe, Ari? Is that what
Yeah,
you're?
maybe a new glucose, right? And this is actually, and again, I don't want to get too much in here, but the reason why a lot of these steps are actually reversible, because there is a reversible process that uses these same enzymes. And it actually has to be that way. In order for you, when you are energy deficient, one of the ways that you can make energy in the form of glucose, because usually it's because your brain really needs it and it only relies on the glucose in your brain, in your blood, apologies, is that I have to have a source of remaking glucose when I don't have other things around. So gluconeogenesis is that process, and a lot of the steps are using the exact same enzymes, but essentially pushing this reaction in reverse. So that's why, if you're interested in this, that's why a lot of these steps are actually reversible. Okay, so let's keep going here. Let's see. So we've done step 7. We're actually going to skip over some of these other ones. This is, if you think about this, well, I'm taking the phosphate from the 3 and putting it onto the 2. This is as a form of an isomerase. And then it's going to take that 2-phosphoglycerate and make it into phosphoenol pyruvate. We're going to focus on this last step. Okay, so this is step 10 here. So as you can see, this is another step where we're going to make some energy. It would also make sense to call this a kinase. So this is pyruvate kinase. And this very last step is the last of, so there are 3 irreversible steps. This is the 3rd of the irreversible steps that you need to know. We generate some ATP. This is a form of substrate phosphorylation. And this is the last of the 3 irreversible steps. So you want to think step 1, hexokinase. Step 3, phosphofructose kinase, and then step 10, pyruvate kinase, are the 3 irreversible steps that are heavily regulated in glycolysis. You want to make sure you know. And interestingly, this is another place where you can feed forward, right? So this is, an earlier component, an earlier intermediate, right, can also go ahead and say, okay, I need you to speed up. I need you to keep going because I need some more energy. It can activate pyruvate kinase here as well. So this is feed forward activation. Okay, so let's zoom all the way out, kind of take a glance at what we've now covered in the last little bit of time together. And I want you to now think of the net processes here. What do we feed into this? We put some glucose in, and then we get a little bit of NADH. And why is it 2? Remember, you got to remember, there's 2 of these things for every glucose that you make. So I make 2 NADHs, and I make 2 ATP total. And this is an important thing, net. So I've seen versions of questions, from, practice tests and things like that, where they're like, well, what is the total amount of ATP that's made? Well, I mean, it really actually ends up making 4, but we talk about the net here because we actually use up 2 before we split the glucose into 2 3-carbon sources. So the net is usually the more important thing, but you also want to remember, yeah, I'm actually putting in a little bit, I'm using a little bit of energy as well. And obviously we're going to make those 2 pyruvates as well. Okay, time for a question. Time for you to think about what we've discussed in the last half an hour. So what I'm going to do, I'm going to be quiet. I want you to take a look at the question, and I also want you to glance at the answer choices and notice, are there patterns that are existing here? Another thing to keep in mind is the word least. So keep that in mind. I'm going to be quiet. And here I want you to be a little bold and try to say your answer out loud, or maybe write it down. Or if you want to, you can also talk to the off-camera instructor that you're currently chatting with right now, and then we'll discuss the answer in just a bit. So I'll be quiet. Okay, Eli, what is, I get a lot of students who are very intimidated when they see least and accept or not kind of questions. What is a common strategy that you would recommend in general?
Two big answers to that, Ari. I'd say the first one is, if the original question is clear enough to you in the first place that you really already have a good prediction of that answer, like this is a process that never involves these 3 things, and you have a very clear prediction, you can jump in and you can try and find that in the answer. But often for a least question, one of the simpler ways, especially if it's a complex question in the first place, simpler ways just to look at the ones that do have an effect. Flip the question over. Which ones absolutely are required? Eliminate those answer choices. And sometimes that will get you right to the right answer by working backwards, by eliminating down to the one that has to be left over.
I think I tend to use that second one, because you know what happens when I forget that there's a least? I'll remember for like A and then maybe B, but then I'll forget there's a least and they'll be like, oh yeah, that must be it. It's C because it fits my question, forgetting that there's a least. And then I'll pick the opposite answer choice. So what I often do, and this is kind of in line with what you were saying, Eli, in that second point, is going through the process of elimination, finding what is going to have an effect and eliminating it tends to work a lot better for me strategy-wise. So that's actually how I would recommend you guys all think about this as well, is rephrase this. Which of the following, if increased, would have an effect on the activity of phosphofructose kinase? So if it does have an effect, it makes sense that it has an effect, then I eliminate it, then it tends to be a little bit easier to work through, at least from my end here. Okay,
what would you cross out first if you had to get rid of one? Because it doesn't say positive or negative, it just says effect. So what would you do first?
I would get rid of ATP. So why would I do that? ATP would be the one I would start to get rid of first, because that is lots and lots of energy. And that's a really good point, because it actually doesn't actually explicitly say what kind of effect this is. ATP being the currency of the cell, right? If I have lots and lots of energy, I'd probably want to suppress the activity of phosphofructose kinase. So I'd get rid of this guy here. Okay, so I mean, if you're following along with how we're kind of working through this, well, we have different forms of it. We've got the diphosphate version and the monophosphate version, which is AMP, and then we've got just the phosphate on its own. Well, how else would we want to do this? Well, if I think about ADP, that's the product of ATP hydrolysis. If I have lots of ADP around, it means I don't have the really high energy version that I need to fuel a lot of my processes. So this would probably inhibit, right? If I can spell, inhibit PFK. Whereas these guys that are the product of the hydrolysis would probably activate it. But in either case, they are both having an effect. So that means none of those latter three answer choices are going to be correct, because they will have an effect. The correct one for this one is going to be inorganic phosphate. So you might be thinking, well, you just told me the product of this hydrolysis is going to have an effect. In one sense, you're right. ATP, AMP, ADP are all forms of energy carriers, but inorganic phosphate is not limited to that. You can see this in like nucleotide generation. It is littered all over the cell. So the amount of inorganic phosphate in the cell doesn't really give you an indicator of the energy need or the energy fullness of a cell. Hence why this is going to have the least effect. So the correct answer to question one is going to be answer choice A.
Now I've got a follow-up question, Ari, because most of the students that I have get rid of ATP because of the inhibition and get rid of ADP because it's a product. So at the very least, Le Chatelier's principle, right? If we've got too much of one side or the other, it's going to go backwards or forwards depending on whether we're talking about product or reactant. But ADP is in the slides that you already showed. It's in the common reactions. AMP isn't normally put into any of these reactions. Can you talk about what's going on if we have AMP, just so we're all on the same page?
So, I mean, we don't usually use ADP, sorry, so adenosine diphosphate, but it's not that it can't be. And so if you think about like where we're kind of envisioning what's going on, the reason why things have energy is when we put things in close, like charges in close proximity together. So ADP, even though it doesn't quite have as much energy as ATP, still has a form of energy and it can be used in other processes. So AMP is like the least energy full version of any of these. And even though it wasn't explicitly mentioned, I want you to think about the reason why that would make sense. If I only have one phosphate on there, there really isn't anything to make it energy dense at all. ADP has a little bit more energy. ATP obviously has the most. So think about those charges. If you're ever in doubt, especially, in a lot of these chemistry heavy versions of biology, at the end, and I didn't know this when I was in school, but I'm realizing this more and more. It comes down to physics. Physics is the root of all of these answers, which kind of makes me surprised, but I appreciate it more than I certainly did when I was in school.
As a physicist, I approve this message. Do we have another one we could do, Erin?
We do. We have one more. And again, I will be quiet. Go ahead and take some time and work through question 2. And I want you to think about those really important steps that we had described a little bit earlier. So short question stem, enzyme names as answer choices. Someone who is a little bit new to the process might be panicking, I feel like, right now.
I think that's fair. I think there's one or two I could get rid of, right? Again, sort of the same method as the last question.
Yeah. And if we're paying attention to our emphasis for which enzyme steps were important, instead of thinking, well, which one does catalyze a reversible reaction, the MCAT savvy way of understanding this one would be like, well, I don't know all of those, but I really do know the 3 big ones, don't I? I know step 1, hexokinase. Oh, look at that, it's an answer. So that can't be it. I know step 3, right? That's phosphofructose kinase. Look at that, there we go, that's B. And then I know the last step, step 10, that's pyruvate kinase. That's the last irreversible step. Ta-da, we did it. Right? So this isn't asking you to have all those memorized for this particular question. If you know that there's only 3 irreversible steps, well, those are the 3 wrong answers then. And so the correct answer is going to be the one that you don't probably know as well. That's phosphoglycerate kinase. So that's one of the ways that this can be tested. Focus on the big picture. Consider the net reaction. Consider how this all fits together and connect the dots. Try not to just throw yourselves into just rote memorizing each one of those steps. I mean, that's definitely one of the ways you could do this. But I will say the one thing that's important for the MCAT is because it's such a breadth of knowledge, you got to anchor that knowledge to something, right? It works for undergrad classes to compress it, every little detail in a short period of time, but you can't keep all of those things in mind without some sort of organization aspect here. And I think metabolism is one of the greatest examples of that action right there, that you have to have this organized in your head. So take the time to learn this slowly and carefully, and it won't disappear at least nearly as fast as you might think.
I think that's a great follow-up point here is knowing those shortcuts, right? These are the 3 irreversibles that sort of shortcut you to memorizing more content than you think you've memorized. Same thing for, we saw yesterday, for those of you here in the optics lesson, there's a lot of shortcuts to understanding magnification or understanding the formula applications that don't require you to memorize literally everything. If you're looking for what are the things that are positive value in a lensmaker equation versus a negative value, you don't have to memorize every single one of those. If you memorize just the positive ones, well, then you've got the negative ones because it's whatever's left over, whatever's not there. So I always love thinking about that big picture view. What are the sort of shortcut ways that I can memorize twice as much as I think I have the ability to, or as I think is available to me. So I'm gonna, if it's okay, Erin, I'm gonna take over from here and talk us through the next big piece. And Erin and I planned this ahead of time. Notice we spent a bunch of time up front going through a lot of the big picture applications, talking about the divalent cations, talking about NADH versus NAD+, where are we reducing versus oxidizing. So absolutely, we are gonna try and, on purpose, move through this one, leapfrogging off of that understanding. That's the whole goal, right? Is that every time we do a subsequent metabolism or a subsequent piece, we already have a little bit of that background. So when we're jumping into citric acid cycle, probably the biggest question that I get is, this looks like it's a cycle. It looks like everything kind of stays in this cycle, but something's got to come out of it. Ari, what's the main reason we have to have citric acid cycle? Like, what is it doing for us for metabolism?
So a little bit earlier, I mentioned how the importance of the electron carriers. And there was such a good question that came up earlier. It's like, what is the role of those electron carriers? Notice how much more of that actually occurs here. And if you think, again, chemistry-wise, if I am generating electron carriers, it's taking those electrons from something. What is it coming from? It's coming from all of that hard work we had actually put into the system from glycolysis. I'm going to grab those electrons from carbon, I'm going to oxidize them, and then make a lot of electron carriers. So while it does, I agree, I thought this was really confusing for a long time. Why is this just like a cycle? It just goes on and on. No, those electrons move and they take off. We've got to insert in more electrons in the form of carbons just as the entrance into the TCA.
Yep. And I love that explanation. And I love that you keep using electrons, because at the end of the day, right, that's the reason why this is here. And it helps us remember, hey, this is happening in the mitochondrial matrix. It has to be, because we have to be pumping those electrons to one side and then pulling the hydrogen through the other side. Makes a lot of good sense that we're ending up with all of these NAD pluses turning into NADHs. I'm actively generating that process. I'm actively making sure that that this citric acid cycle is actually doing the energy transformation necessary. I want to make NADH. I want to make FADH2. I want to make some GTP and some byproducts. We'll talk about these CO2s in a second, but that's the whole point of this process. So let's start with getting into the first part of it, because we can't be in citric acid cycle from where Ari left us a second ago, unfortunately. So we've got pyruvate, and we've got to make it into acetyl-CoA in order to get into the citric acid cycle. All right, great. I've got to make pyruvate into acetyl-CoA. Well, I know that there's a hydrogen that gets pulled off. I know there's also a CO2 that gets taken out. This is a big complex. Actually, this is a mix of several different enzymes that get lumped together in order to make this happen. So generally, when we talk about this, we talk about it as a pyruvate dehydrogenase complex. It's a couple different things all lumped together. Technically, there's 5 enzymes lumped together in the complex. At least 3 core coenzymes are involved in it. So at the end of the day, our goal is just to make sure that we get acetyl-CoA out of the end. Now, I'm always interested in where we've got carbons and where the carbons are moving around. So I know that I started with my pyruvate over here on the left. I know this is in the mitochondrial matrix because I have to have it happening in there to move the electrons around and gain the protons back. But if I'm generating NADH and I'm producing that acetyl-CoA, I also care a little bit about when am I going to actively want to push this forward versus when am I going to stop this process? Like when do I have enough acetyl-CoA? But before I jump into that, it seems like this is necessary. I have to get acetyl-CoA. Ari, is there any other way I could possibly get acetyl-CoA outside of glycolysis that you just left us with?
That's a great point. I've always described acetyl-CoA as the hub. This is like the center of so much of metabolism. So this isn't the only place you get acetyl-CoA. A common way you can get that actually is from the breakdown of fats. So lipolysis and then cycles of beta oxidation is another very common way. You can take little 2-carbon chunks off of a big fatty chain and then make a ton of those. You add them into the coenzyme A and it now makes a lot of the acetyl-CoA. So that's one form. It's not the only one, but it's a very big one.
Absolutely. Fatty acids, ketone bodies, alcohols, right? Anywhere I can lump off those little pieces, I can make acetyl-CoAs. Now, let's think about that feed forward, feed backward sort of a rundown. So we've got a 3-carbon pyruvate and a 2-carbon acetyl-CoA. Ari, you and I were talking about this before class. I have memorized that acetyl is a 2-carbon thing. But what's the big question that that we seem to always get?
Why is acetone a 3-carbon thing when this is a 2-carbon prefix? Tell me why.
Yeah. And unfortunately, this comes from some old nomenclature. So we have IUPAC now, and IUPAC is very, very good about exactly the number of carbons. So if we're talking about acetone, right, classic solvent, comes up tons across chemical reactions. If I talk about acetone in IUPAC, it's propanone. Now, we don't even have to say 2-propanone, even if your organic chemistry professor in college said you have to number every single one. You don't even have to, because there's only one place it can go. But that is the crux of the problem with acetyl-CoA. When they were naming the structures, they, back when they didn't have IUPAC, they named the simplest possible thing I could make. Usually it only has one carbon. They named it form to be like the simplest thing. So formaldehyde, formic acid, et cetera, is the simplest possible one you can make. Formaldehyde is just one carbon. And the second smallest thing they would call acete for second smallest, so 2 carbons usually. So acetaldehyde, 2 carbons long, check, check, check. But when they got to acetone, they were able to extract that, they were able to analyze it in certain ways, and they were like, this is way too big for this to possibly be the smallest version of this molecule. It must be the second smallest. It's got to be the second smallest ketone. So acetone is, of course, the absolute smallest. So really, if we're going to go back in time 100 years, we should go back and rename it forminone. And that I think, this is something we could stand for. We can pick it for this, Ari.
Forminone it is.
Or we could move on. But that, if you were all confused about this, about why this is a 2 carbon thing when acetone is a 3 carbon thing, but then acetic acid is 2, and et cetera, et cetera. That's the reason. Acetone is just the random example where they got it wrong. All other cases, acete does mean 2. Now, let's jump back into the feed forward. Why would I push this forward? I'd push it forward in low energy states. And of course, I would have negative feedback against it if I already have too much acetyl-CoA from things like my fatty acids or ketone bodies, or if I already have plenty of ATP. Again, Ari made a big deal about this earlier, and I absolutely want to mimic that. Anytime I have too much of that final product, especially if I have NADH, this is highly regulated across that membrane because it is part of that electron-specific carrier mechanism. If I can't regulate that, or if I can't use that for regulation, I might end up with too much product, and that causes a whole bunch of downstream problems. The only other thing that I'll mention for this is that this is taking a 3 carbon to a 2 carbon group, but the reason we have to have that in the first place is because I have to meet with someone else in the citric acid cycle already. It's a cycle. It's going round and round and round and round. So I have to get to something that can then bind with oxaloacetate, because that's the end of this loop. I guess we should look at that again. So here's that loop again. Now I've got my acetyl-CoA. I've slid right in here, and oxaloacetate is the last thing I ended with. Now we can begin the cycle itself, if you want to think of it that way, or we can continue the cycle at kind of technically the first step. There's never a first step in citric acid cycle because it's always running. First step happened way long ago before we were ever around to even see it. So now if I want to make citrate, gosh, if I was going to name an enzyme, Ari, to make citrate, I mean, I could pick all of the possible options available to you, but what do you want to call it?
I think it really should be Ari synthase, but nobody's going to go for Ari synthase. I really want to have an enzyme named after me, but I know it's not going to happen because biochemists are generally not all that greedy. So let's call it citrate synthase then.
All right, we'll go with it. It's really the organic chemists. That's, Claisen condensation, Wolff-Kishner reduction. They get all the good names. So here's our first step. We've got the oxaloacetate, again, 2 carbons in acetyl-CoA, 4 carbons in oxaloacetate. And we want to do something with it to make it into citrate. Citrate is going to be my 6 carbon group, but it's a 6 carbon group, and acetyl-CoA and oxaloacetate both have carbonyl groups in them. And I want to end up with citrate. Citrate, remember, citrate, the ATE ending, that just means negatively charged stuff. Like a carboxylic acid group becomes a carboxylate group just by losing the proton. So citrate really already tells us this is a negatively charged something that's floating around in space. If I were to put the protons back on it, citrate, I would guess, would probably be citric acid. Would make a lot of good sense. So knowing that background organic chemistry can actually help you out in places like this. So here's my 2 carbon, here's my 4 carbon, and I'm going to push it over here into the 6 carbon that, you know what, now that I look at it, does look pretty darn close to citric acid. I have a 3 carboxylic group regions. And again, the whole point here being, I want to understand a little bit better what I'm looking at, what happened in each of these reactions. So I've made a 6-carbon group. This should still be in the mitochondrial matrix, but because I'm putting two things together, it's a special class of reactions. So this is a condensation reaction. Now, it might be a silly question, Ari, but this reaction looks a little bit complicated. Do we think it's going to be reversible or irreversible?
It's a great question. Well, I mean, if I'm grabbing 2 carbons from acetate or acetyl-CoA, sorry, and then I'm going to add it into the oxaloacetate, it kind of looks like I put a bunch of, I don't know, negative charges in close proximity again together. I'm guessing probably not.
Yeah, right. This should it better be irreversible. I really don't want this to be able to slide backwards. And in general, if we make something with a big carbon chain in it, or if we knock off pieces, spoiler alert, from a big carbon chain, that sounds like a situation where in order to go backwards, in order to, in this case, reverse the condensation reaction, I would really require a really complex structural conformation change to get all the way back to those first 2 groups. And again, to your point, those first 2 groups are already negatively charged and require a lot of additional oxidation to happen at each of those subpieces. In this case, I've made my final product. It's a more stable final citrate molecule. And that's what I want to have stay. So irreversible reaction for sure. Feed forward if I'm lacking energy. Again, ADP should make in your head, lack energy, I need more. And then I want some negative feedback if I have too much energy, so too much ATP. Now, you see in here succinyl-CoA, which again, if we've got this product that is kicking off the CoA groups, I'm ending up with a lot of extra residual product that could be part of the succinyl-CoA, which we'll come back to in a little bit. But that's part of a subsequent reaction that's actually feeding back all the way here. I like how, Ari, when you were talking through glycolysis, you kind of talked about the energy input steps and then the energy payoff steps. I think the same thing is true for citric acid cycle, but people don't generally think of it this way. You know, the first couple steps of citric acid cycle is the setup. You know, get the pieces in place, chop down the molecule to the right size, and then the back end of it is the recycling, you know, getting ready for the next piece. So if we can set it all up and then do that one little energy transfer, then the back half is the recycling. So we're still in that setup, still in making sure we have the right molecule to start with. So now we have citrate going to isocitrate. All right. Citrate converting to isocitrate. Now, this sounds like I'm just moving around positions of some kind. I've got a question. We kind of skipped over the mutase in glycolysis because I agree with you. Those steps are not like hugely important for us at a structural level. Just like the isomerase to get from glucose to fructose is just moving that one around to make the reaction go a little bit better. But I think it's worth our time stopping here for a second and thinking about this reaction from a organic chemistry standpoint. So what's the benefit to taking a tertiary alcohol and making it into a secondary alcohol for a reaction viewpoint?
It's a great point. I think one of the key things that you want to keep in mind, because really this is just chemistry. It's chemistry that happens in your body. I really think this is a lot more understanding if you know what comes up next. And the next part of this is oxidation. So when we are oxidizing things, again, for the purpose of pulling electrons off to put it to our electron carriers, you can't really oxidize, you know, like carboxylic acids are about as oxidized as possible. The only thing you can oxidize is that alcohol. But when it's in citrate, that alcohol is on what we call a tertiary carbon. So tertiary carbons are carbons that have no other hydrogens there. So if I wanted to oxidize a tertiary carbon, I can't. I don't have any hydrogens to kind of kick off to be able to oxidize and turn into a carbonyl. So instead, what happens in this actually really, really important step is it moves that hydroxide group from the tertiary that's in the middle to the one in the bottom, which is now that secondary alcohol. So that is now ready in preparation for the step that comes after it.
Yeah, I love it. A super detailed answer. Again, if you ever get a tertiary alcohol on the test anywhere, and they say, which of the following is most likely to be a product from it? Tertiary alcohols don't like to oxidize. They don't like to do anything else because you can't. If I tried to oxidize that one more down, make it into what, a carbonyl, a ketone, or an aldehyde if it's on the end. Well, couldn't be on the end, right, for a tertiary, but a ketone or a carboxylic acid, like you said, or make it more oxidized, impossible. If I'm tertiary, it can't be an aldehyde. If I'm tertiary, I can't make a ketone. I would have a pentavalent carbon. Or I'd have to have kicked off a carboxylic or something, which, again, can't do it, never going to happen. So tertiary alcohols, even if you have potassium permanganate or something super strong like an oxidizer, still never going to do anything. We had to make it into a secondary alcohol for that to happen. And that's why I didn't click the last click on this slide, because that's the whole point. The whole point is creates a more easily oxidized alcohol. I don't even love the word more. Cross that out completely. An easily oxidized alcohol. We couldn't have oxidized the first one. Impossible. So now, absolutely, let's jump back to our big picture view of this and take a look at making that isocitrate into alpha-ketoglutarase. So step 3, I think from a big picture point of view, what I would say about it is this is producing NADH for the first time in the process, first time within the cycle. So we're going to convert it into technically to an intermediate before we make it into alpha-ketoglutarase. As Ari said earlier, and I'm just going to parrot again, a lot of the intermediate steps are helpful for some understanding at a biochemical level, but generally aren't tested specifically for that step. The big picture view is we get to alpha-ketoglutarate. Now, in order to get to alpha-ketoglutarate, I lose a CO2. So we can think of this first step, the intermediate, as being something that was just necessary to happen, right? Isocitrate dehydrogenase is going to catalyze the oxidative decarboxylation next. But that decarboxylation, the CO2 leaving, I'm curious about this. CO2 is leaving. Does CO2 just leave and hang out nearby in case it has to go back on?
I know CO2. CO2 is not a very happy molecule in aqueous environments. That's not going to hang out there at all. So it's gonna, we're gonna respire this eventually. So yeah, no, it's not gonna like hang out there. That means actually something pretty important about this step, right?
Huh. Now I know that we're gonna, it's still in the mitochondrial matrix. We haven't left the mitochondrial matrix. The whole thing is in there. And I did already say it's the first step in NADH. It's the first one that made CO2. But as Ari was hinting at, it's got to be the first step that is absolutely within this process, besides the first step making citrate, that's irreversible. So we're still in the build-up process. We're building up the thing that's going to make that conversion for GTP. But this is one of our irreversible steps just because CO2 got lost. And I hinted at this earlier. If we throw together, we do a condensation reaction for carbons, that's an irreversible reaction. And if we lose CO2, we've lost a carbon piece. It's floating out there. We're going to expire that. It's absolutely not going to be sticking around to go backwards. It can't be reversible. Now, thinking about those forward reactions, again, we have manganese listed out here. I know we talked about this earlier, but I'll just say it's absolutely possible for us to use magnesium for this reaction. It's another divalent, but under physiological conditions, manganese is the cofactor that generally gets used. I always like to point it out that, hey, this is different from magnesium, which we were talking about all the other processes so far, or calcium that we see other places, or iron, right? Iron 2+ can sometimes be used for these reactions. The key piece is divalent. So is the MCAT going to say, hey, do you know the one reaction that physiologically is usually manganese, but could actually be magnesium? If no, that's too specific, too beyond the scope. And I hope that secretly answers a bunch of people's questions that have been furiously writing out notes as we go through these, that a lot of what we're covering is big picture, where we're using specifics to really drive it home. But at the end of the day, these should feed through the entire process. So what do I expect to make this reaction go forward? ADP, NAD+, or maybe something that means we're metabolically active, so like a calcium, something like that. What would hinder this reaction? ATP, NADH, right? Too much of my product is going to hinder this and make it go backwards, or at least hinder it from moving forwards. So because it's irreversible. So as we're thinking about these, I see a lot of questions about how to connect these to other processes. What is the number one set of these? Like, do I have to know the pentose phosphate pathways? Some of those do come up. Certainly glycolysis, citric acid cycle, way more commonly than the rest. But even in those other processes, we can use this big picture view. What do we need to make? Where do we get our energy out? And what would move the product forward or potentially be a negative feedback? Now we're still in the build-up phase as far as I'm concerned. We're down here at the very bottom, getting to succinyl-CoA. And I've got to do something here. If I was going to talk about this as a process, it sure looks like I lose CO2. So this looks like it's an oxidative decarboxylation. I'm losing that again. But much like the original citrate pathway, we have a whole complex of stuff that's actually necessary. And it's actually really a multi-enzyme complex using several of the same ones as we used up here in PDC to get acetyl-CoA, which I know is a favorite trivia extra note of Aries.
I love this fact.
Bonus fact. Now, again, the multi-enzyme complex, even if I don't have all of those little pieces memorized, I assume I'm going to have a divalent cation. I've got magnesium here. And of course, this is still mitochondrial matrix. Nothing has changed there. We're losing CO2. Again, I know we're going a little quickly through this, but it's on purpose. I hope you're seeing patterns. As soon as I saw losing CO2 in my head, I changed that and I said irreversible reaction. So it's the second step where we lost CO2. It's the second step where I generated NADH, and absolutely an irreversible forward process. Now, I don't have to spend an extra minute talking about the fact that this is promoted by ADP and NAD+, and hindered by NADH and ATP, because that's what the whole rest of them have been as well. So I hope that as you're seeing this, you're saying, oh, reaction regulated in a similar fashion. Okay, so for creation of these products, I'm expecting that same sort of a mechanism. And because this is the buildup, this is the place that I have to have inhibition, or I have to have feed forward, because this is the part that I want to regulate. I said at the very beginning, but if we divided citric acid cycle in half, it would really be building up the stuff that I need, or in this case, slicing off carbons at a time to get the thing I need. Once I've done that, whole back half is recycling. So I don't have to directly promote or hinder the back half of this reaction. That's going to be all the recycling steps. I do have to have these for the lead up steps. Now, luckily, we're here at the actual step where we get to do something a little bit interesting, where we get our final product, if you want to think of it that way. So our final product here, this is where we're actually going to create something useful. It's not ATP, it's GTP. Eric, I always get asked about GDP to GTP. Isn't this an energy generating step?
Absolutely. Similar to how ATP would work the same way, ADP to ATP. The only difference between these is the nitrogen space, right? This is guanine instead of adenine. Functionally, though, the cell considers them energetically the same. So I don't really want you to think, okay, well, I got another random fact I got to memorize. GTP, from the point of view of the cell and requirement of energy, it's really the same idea. This is a way of generating ATP, but not in the ATP synthase, right? Not in the electron transport chain.
Yep, love it. That's exactly, and I usually use that same idea. Right? This is a substrate level phosphorylation, right? It's happening at the substrate level. We've got that energy transfer, and we're directly making GTP. So GTP generation specifically. Now I've made GTP, right? My analog for ATP. I've made the only piece of energy that this entire loop is going to do. Again, citric acid cycle is not really designed to make energy directly. It's just not very efficient at it. It's designed to move electrons. That's what we care about here. We're in the mitochondrial matrix. We're moving electrons around. We're moving protons around because the mitochondria, that's what's going to make our energy. That's where the big money comes in. So this is just loose exchange of electrons, loose exchange of protons. Yeah, we're doing a little recycling here because we don't want to lose that delicious pyruvate. We don't want to waste any acetyl-CoA. We want to make sure that mitochondria gets the energy that it needs and gets the waste products from this essentially that it needs to actually make the big bang ATP. Well, so now I'm halfway through, right? Now I've got to be recycling. The back half goes pretty darn quickly at this point. Okay, if I'm recycling this stuff back out, I have succinate, I'm going to make fumarate. So I have to convert it to fumarate, but notice the big product here. This is the only place where I'm really, really, really pushing a lot of hydrogens around. Yeah, we did it with NADH as a product, but now I have FADH2. So I have extra here. As far as the process, this reaction is kind of unique because the enzyme itself isn't actually directly in the matrix. It's part of the integral membrane protein in the inner mitochondrial membrane. So it's covalently linked and contributes back into the electron transport chain directly. So again, I'll say that again. It's not in the matrix, even though, yes, this whole thing is in the mitochondrial matrix, but this is on the inner mitochondrial membrane. So what we're doing here with this succinate dehydrogenase, or SDH, is a membrane protein on that inner mitochondrial membrane. So covalently linked to SDH, right? And that's what's allowing us to make this FADH2 so so robust, or so much hydrogen transfer in this step. Now, I did see a question come through the poll version of the chat, and I know that a couple of our off-camera instructors have been answering a bunch of this, but we keep talking about dehydrogenase. Area, if I know that the product from this was FADH2, what do I know? What do I say happened to succinate here in the first place?
Great point. If I'm making something that is carrying electrons, the electrons again has to be coming from something. That means that succinate then must be oxidized, right? Remember, redox must always occur in pairs. You're not pulling electrons from the thin air, right? It has to be coming from something. Where's it coming from? It's succinate. I also think this one process made a ton of sense to me when I'm like, wait, where have I seen the movement of and the generation and the usage of FADH2? You know, Eli pointed out that this is on the interchondrial membrane. I also want to point out the fact that the other name for this enzyme, the other name for succinate dehydrogenase, is complex 2. That is the actual link between the TCA and the ETCs right here. Because like while all the other electron carriers, which I mean, the only other electron carrier, which is NADH, kind of does a little bit of migrating and moving around while it floats around with this electron. FADH2 is the only place where, as it's made, it's almost directly used to give up to CoQ, actually.
Yep, I love it. Good connection. And again, we're recycling here, so let's keep recycling that fumarate, make it into malate. Again, the malate formation process should make a lot of good sense in that I'm essentially taking something that I just formed and I create that water reaction across, again, in mitochondrial matrix using water. But if I'm putting water through a process and I'm using an enzyme to hold it in place and lock it down, I know what water had to have been applied to. There's only one thing that really works well to hydrate across, and it's either one of two things. It's either we've got some sort of internal ether, internal ester, where I'm hydrating across an oxygen link. That's a great place to add water across, because then I can get two alcohol groups off of where I originally had an oxygen bound. Or I'm using water across a double bond, because putting water across a double bond puts a hydrogen on one side, hydroxyl group on the other side. And because I don't have any internal oxygens, I know we must have had this added across a double bond in fumarate to make malate. Again, just trying to make it make a little bit more sense so you're not just straight up memorizing each of these pieces. And I'll remind you, we're in the back half, so that's all recycling. But all recycling, it's all recycling and oxygen add, right? So whenever possible, we're oxidizing, oxidizing, oxidizing. And in the pair to that is we're always creating that NADH. if we're oxidizing each of those things as we recycle it, to oxaloacetate, which, gosh darn it, sounds like it must have the most oxygens if we're going to call it oxaloacetate. Yeah, absolutely. One more step to make oxaloacetate reformed at the very end. We take our malate, we add one more possible oxygen connection. Again, this had better still be in the mitochondrial matrix. Final step, regenerated. Now we're looping back through. So first half build up, back half recycling. Let's take a big picture view of this whole thing. So here's my big process from the point of view of energy. If I lack energy, if I'm running out of it, I want to make sure that I feed it forward. If I have too much, so I've got plenty of ATP, I want to shut it off. But I want to shut off the first half steps. These are the things where I'm building up or losing CO2s, but creating that process to give me GTP. And if I already have enough energy, I don't need to do any of those. Again, if I can hinder those first parts of this process, I don't even have to worry about hindering the recycling steps in the back. I've already hindered this so much, it's not going to even make a difference, those last pieces. Same thing in reverse. Again, nothing fancy here, but if I lack energy, ADP, I better promote all of those initial steps. So I like to think of glycolysis as really being this, this input-output process for ATP, and citric acid cycle as, again, sort of an input-output process. The first half is build up, the back half is recycling, and at the end, the thing that we've been talking about from the very, very beginning. We are netting out, yeah, a tiny bit of GTP, sure. We're netting out a tiny bit of acetyl-CoAs being used in the first place. But at the end, we're really making NADHs and FADH2s. So let's put that up here on screen. Here's our net used versus made. Oh, and there's our big picture view. I like that, that big picture view because NADHs and FADH2s, as Ari already said, enter the electron transport chain for oxidative phosphorylation, which, you know, would be the next topic if we were jumping to it. But I think do we have time, Ari, you think, to do one question here at the very end?
Let's do one really fast one.
You want to do the hardest one?
Yes.
I like to do the hardest one. So let's grab this one. This is the hardest one by far. This is the one that we tend to see a lot of students, unfortunately, make mistakes on. So let me be quiet for a little while, just so you can even just read through and get a sense of what we're doing in this question. All right, now I'm sure that's not enough time to fully do this question, but if it is, amazing. You've figured out a shortcut on this one. There's a lot of ways that I could explain this, and the slowest possible way is probably to go back and pull up a picture of the Krebs cycle, of citric acid cycle, and look at oxaloacetate and see, like, what would I need to do in the forward loop to get all the way around to succinate. But there's no irreversible process between those two pieces. I don't have to go forward in the citric acid cycle. I could go in reverse. There's no reason I can't. Those are not irreversible processes. So one way to do this question is to pull up citric acid cycle, Google if you don't have it handy, and then just trace it backwards, and then see what would have been made in the reverse of this process. That's fine. I think a simpler way, and this is what Aaron and I were talking about before class, is to look at what happened to the molecule. Oxaloacetate to succinate, it only changed in that we had a carbonyl here. Makes sense, right? Oxalo has the most oxygens, and it turned into this, which has a hydrogen there in its place. Well, in order for that to happen, I had to have pulled this group off and replaced it with a hydrogen. I need to have burned something with hydrogen. So it can't be that we produce NADH in this process. It can't possibly be A. There's no way I can produce that if I have to have used it. So let's get rid of A. Let's get rid of D. I'm not producing NADH. If anything, I had to use it to put a proton on right here. Well, I guess the second piece then is ATP. Is this part of the pathway where I had to use ATP to do it? And again, if you thought of the whole forward loop with all of those, you might have thought, well, it could use GTP. Maybe that's an analog. Maybe it's close enough. But absolutely, we could go in reverse. And if we're going in reverse, there's no GTP. There's no ATP. There's no reason for me to be burning any energy. So I will not have any of that happen. I will not have it consuming that energy. There we go. Let's just delete my squiggles. So it can't be consuming ATP. It's got to be that instead does not consume ATP and produces NAD+. No possible other option here. I love when we can get a question that is super complicated that, again, I could pull up citric acid cycle, we can go backwards through the entire process, or we could just think of it from a biochem standpoint, shortcut a lot of the work because we recognize from the very beginning that this was all about oxidation and reduction. So again, we've gone through a ton of stuff here in this session today. At this point, it's always nice to kind of think about this from the big picture. So I encourage you go back through, look at this at a big picture. When you have access to the recording within 24 hours on our YouTube channel, come back and watch it through again. But Ari, I know that we always like to take a little bit of time at the end, and we don't have a ton of time. But I would love to know sort of your big tips for, what do you usually recommend to students who are working on studying this heavy, dense content over the course of weeks and months?
Certainly not something you can do on a whim. I think you really need to make sure that you're planning, like really put this into perspective. This is 2 years worth of undergraduate courses compressed into one test. It's not something you can cram. It's not something you can do just for like a couple of weeks and then just wing it. I really want you to think about making a plan. And I would say like that's the #1 question I get from students is how do I make a plan? What do I start with? First, gather resources. Think about like what resources do I have? Do I have practice questions to use? Do I have the right books? Tap into expertise. Like think about friends who've already, successfully gone through the MCAT. Come to events like this and ask us questions. We'd be more than happy to talk about, what are some of the, bigger tips that you would advise or we would advise that would be good to consider. Like thinking about your time right now. Like, are you in school or do you have a job? Do you have like a lot of busy family life? Think about your commitments and think about, is this right now at this moment in time a good time for me to be tackling a test with studying like this? So I think like all of that takeaway, plan it out. Don't just like do it halfway. Make sure that you are appropriately and realistically planning this out for a specific testing.
Yeah, I think that's a great point, Ari. I love that you said, planning out your time and like tapping sources of expertise. I like to think of the MCAT studying as an exhaustive process in general for most people, but we can make it more efficient. Actually, that's how this session today got designed, was around this idea of what are big pain points that we can streamline and make simpler, make more efficient, so they're not just exhaustive with you spending hours and hours of your life outside of class trying to learn it. I don't know where we got this bum from who's teaching, it looks like kidneys, but I hope he did a good job on that session. But the MCAT channel is one of the things that we designed around that, where instead of it being an hour and a half like today's session, we have 1 hour, very streamlined, focused sessions that are the core material you have to know. Again, looking at all the patterns, like what Ari and I went through, but for, in this case for kidney, in this other case for molecular genetics, so translation and transcription. So really narrowing down to what gets tested, how to memorize, how to learn it, even more than memorize it, so you can get those bigger questions on the test. And Ari, you already mentioned, streamlining for what you need the most. I think that's one of the best things that comes with all of the resources that Kaplan offers, be it studying on your own, be it doing a whole class or doing tutoring, is that you really do get that the background of where is your best place to focus your energy. I'd love if everybody was already at a 517 focusing their energy like this example student, but a lot of times you're coming in and you don't know where you are. So you need practice in the first place. You need something to tell you, how can I be more efficient? Now, I've got one other question that I saw come through a couple of times in chat for you, Ari. Not as many for me, but I'm not jealous. How can they see you again?
I have a class that is coming up. I'd love for everyone to join me in a live online class. So if you like, you could copy in that link and you can take a look and see the schedule. It does start in a couple of weeks. It's kind of late, but I will say it is perfect for Pacific Coasters. I'm personally at East Coast, which means it is definitely late for me, but I love the nighttime courses. Get a lot of really good chatter there. So feel free to look up options like this one or any of the other upcoming options. Eli, as you saw in that little picture, does appear in the channel very, very regularly. I love working with Eli. He's got some of the best mnemonics and tips and analogies and really great ways of talking through content. And you can also look into options there as well.
Yeah, awesome. I think the last thing that I would say about studying or practicing or trying to figure out the best way to maximize your resources is there's no bad way. There's no way you're going to fail studying. You're going to put in, on average, an MCAT student puts in, 300 to 500 hours studying for the MCAT, which sounds like a massive number, but it makes a lot of sense. And you are efficient. You know that you can be, an alpha student, a gunner student in general. You wouldn't be on a pre-med track if you weren't. The key piece here is really just trying to be efficient and not waste your own time. So to that end, I know Ari has this class coming up. I'm jealous that I'm not teaching it with her, but you might not want to dive right into that class right now, or you might want a more leisurely plan, or you might want tutoring, or there's so many options out there and so many different packets of resource you could be considering. I really encourage, if you're interested at all, if today's session was fun, check out this, the MCAT homepage.youcanbook.me. That's a way for you to directly pick a time slot to have someone, one of us, reach out to you and say, hey, here's where it sounds like you're at. Here's what, from my professional opinion, teaching for the last 15 years, this exam, here's what I think would fit best for you. And I don't recommend the same thing for every person, every situation. There's a lot of variation. I mean, we've got dozens of different options to fine tune and pick between. So that's the last thing that I would say from my standpoint is the thing I dislike the most is when a student comes into a class where they're going to spend 300 hours studying and they've already, I won't say wasted, but burned through 100 or 150 hours by doing something not efficient or trying to scrape together videos from somewhere and practice questions from a 5-year-old book they got from some friend who took it a long time ago, and just weren't as efficient as they could have been. Any last words from you, Ari?
I can't echo that anymore. I think we want to make sure that your prep is customized to you. This is a big time investment. This is the start to a lot of people's dreams. And I don't want you to waste your time. So please come talk to one of us and tell us a little bit about your background, where you're at, what your goals are, what your test it is, all of that information. We'd be more than happy to help you and figure out what would be a best plan for you. Like Nick mentions, it's not cookie cutter whatsoever. And that makes me always the most sad is when people are like, well, here, this is the only thing I've got. No, not at all. It's not the only thing. And let us help you. Let us help you figure that out, because it's a kind of a complex task. And we've heard really, I think, every variation story between the whole host of teachers that we have. We really have heard it all. So please have discussions, have conversations, look into your options. And I do hope, come check us out. I think we have another one on cars tomorrow. Eli and I will actually both be off camera answering questions and stuff. And come check it out. Do another cars lesson with us. We'd be happy to see you guys all again tomorrow.
Well, before we close out, Ari, thank you so much for taking your time and hosting this session with me. I had a ton of fun, and thank you everyone for coming. We really appreciate that you spent the time, that you put in the effort to be here in the first place. It's that sort of effort that does lead incrementally to the success you're looking for at the end game here. So thank you for coming. Thank you for attending. We hope to see you down the line, either classes or tutoring or other free events that we have. And in the meantime, have a great rest of your evening. Good luck studying.
Bye, everyone.